Hi class, this is Dr. A here again, and we are going to talk about metabolism, and we are going to concentrate on two major energy states. One is the absorptive state, meaning you've just had a meal recently, and the other one is the post-absorptive state, meaning you haven't eaten for a while, say 4 or 5 hours. So here are our learning objectives. We want to talk about the absorptive state. What's going on with carbohydrate, protein, and fat metabolism during the absorptive state, also called the fed state. We want to identify what's the major energy source for most of the cells of the body in the fed state. Then we want to go to the post-absorptive state. What are you using? What are you doing with your carbohydrates, proteins, and fats? What's the major energy source for cells during the fasting state or the post-absorptive state? We want to make sure we know what glucose sparing is. And then we want to do some comparing and contrasting of the two states. And then, of course, these processes are regulated by hormones and what's going on with insulin and glucagon and growth hormone and catecholamine and cortisol and their permissive effects and all that sort of thing. Alrighty, so we have two main states. Absorptive state, also called the fed state. You've eaten a balanced meal recently. The post-absorptive state, also called the fasting state, and it's been at least 4 hours since you've had something to eat. And those processes are summarized here. So in this orangey kind of background, that's the absorptive state. The absorptive state, we're building things. We just ate a meal. So we have amino acids that we're turning into proteins. We have glycerol and fatty acids that we're storing as triglycerides. And we have glucose that we're storing as glycogen. Most cells of the body are burning glucose because it's around. Glucose is a wonderful energy source. Cells love glucose. You can just whip it right into glycolysis and get things started and make energy. And what's your liver doing? Well, extra glucose that you have sent to the liver to make glycogen for storage for later, and fat because your glycogen stores are finite, and fat is actually you can store more calories per weight using fat molecules. Now, what's happening in the post-absorptive state? It's been more than 4 hours since you've eaten. So some of those proteins you're going to break down into amino acids. Now, why would you want to do that? Well, you need to make new glucose. And you can take triglyceride and break it down into glycerol, but glycerol isn't going to handle the problem. So you're going to have to use some amino acids for that gluconeogenesis the liver is going to be doing. Free fatty acids are going to be broken down from triglycerides, and glycogen is going to be broken into glucose. Most cells of the body are going to switch to fat metabolism, and that could be burning fatty acids or burning ketones. And those are going to be taken up to cells, and most cells are going to be using fat. Your brain's probably going to be using glucose because your brain really likes glucose, particularly the higher levels of your brain. There are parts of your brain that will run on ketones if you say they have to run on ketones, but glucose really is a preferred energy source for your brain cells. So how are you going to do that? The rest of the body is going to run on fat, and we're going to make glucose for the brain. And how are we going to do that? We're going to perform gluconeogenesis using pyruvate, lactate, glycerol, and the gluconeogenic amino acids. What do they have in common? They're 3 carbon molecules that can be joined together to form a 6-carbon molecule called glucose. Now, here's a fun diagram of the absorptive state. Now, how do I know that? Well, it tells me to begin in the gut. So, I have had a balanced meal. I've got glucose in that meal, as well as some galactose and fructose. I have triglycerides in that meal and amino acids in that meal. Amino acids and glucose first are going to go to the liver. Amino acids Some of those amino acids will be made into proteins. Remember, think about all the proteins your liver makes. Albumin, alpha and beta globulins, those kinds of things, all the blood clotting proteins, all that kind of stuff, angiotensinogen. But the liver actually runs on amino acids. The liver deaminates amino acids, and those alpha keto acids are actually an energy source for the liver. That glucose is also going to go to the liver. The liver is going to take that glucose and make it into glycogen. Some of that glucose is then going to get sent on to other tissues, but anything excess is going to be turned into fat. Keto acids can be made into fat. Glucose can be made into fat. Once you get fatty acids and glycerol, you can make triglycerides. It is very easy for your liver to take excess stuff and make it into fat. Packages it as a VLDL and sends it to the adipose tissue. Now, any fat that you ingested is packaged in a chylomicron, and those chylomicrons are then first sent to the lymph, then sent to the venous blood, then sent throughout the body and to be delivered a lot to the adipose tissue. So in the capillaries of adipose tissue is a wonderful enzyme called endothelial lipoprotein lipase. It's going to break down triglyceride into fatty acids and monoglyceride. Those can then diffuse into the adipose cell, and the adipose cell turns around and takes that and puts it back into triglyceride. Why is that? A triglyceride is too big to get into the cell, and by breaking it down into its components, it allows it to fuse into the cell. You make it back into a triglyceride, so you maintain the inward concentration gradient for diffusion of the component parts. Glucose is also going to be taken up into triglycerides. Adipose cells will use the glucose to make ATP. Excess glucose gets turned into fat. Glucose is going to go into most cells of the body and be turned into ATP. Notice that glucose goes to the liver first. That is a beautiful thing because after a meal, the hepatic portal vein is just flooded with glucose, but the liver gets first crack at that glucose, so then the rest of the cells of the body are not exposed to essentially toxic levels of glucose. So glucose is going to go to the muscle. Amino acids are going to go into the muscle. Muscle's going to use glucose for energy. Extra glucose goes to glycogen. And muscle is always repairing and replacing its protein, so amino acids are going to be used for protein synthesis. This is a diagram from another textbook, Kopsted's Pathophysiology. So we're in the fed state. We've got lots and lots of glucose. Glucose is going to go into brain cells. Glucose is coming from the GI tract. That high glucose tells your pancreas to make insulin. Insulin is going to help get glucose into the muscle, glucose into the fat, glucose into the liver. Liver's not going to use the glucose for energy. It's going to store it as glycogen or fat. Adipose tissue is going to use the glucose for energy. Excess goes to fat. Glucose goes into the muscle. Muscle uses glucose for energy, but also makes glycogen. So you eat a meal, you put a bunch of glucose in the system, and the body either uses the glucose or stores the glucose. Hopefully you can see that's the basis of a negative feedback mechanism. Now what's happening in the post-absorptive state? It's been more than 4 hours since you've eaten, and so you got to keep things going. So we don't start in the gut. We start out in the periphery, where the blood glucose level is in danger of going down. So glycogen is going to be broken down and put into glycolysis. Now, pyruvate or lactate could be sent elsewhere or could be used in the muscle. But the glucose can't leave the cell, but lactate or pyruvate could. Protein can be broken down and amino acids can be sent elsewhere. Adipose tissue can be broken down, glycerol sent to the liver, fatty acids sent to most cells of the body. Most cells of the body are going to take fatty acids or a byproduct of fatty acid metabolism, which is called a ketone, and used as energy. As most cells are using fatty acids, glucose is going to be spared for the brain, hence that term glucose sparing. Now, how are we going to get glucose for the brain? A couple of ways. Glycogen is going to be broken down. The liver can phosphorylate glucose 6-phosphate. Glucose then can be sent into the blood. But glycerol, lactate and pyruvate, and some keto acids can be used to make glucose through a process called gluconeogenesis. The liver can take fatty acids and break them down and make ketones. Ketones then could be sent to other cells of the body in addition to fatty acids to provide energy. So most cells convert to fat metabolism, sparing glucose for the brain. No matter what, the liver uses keto acids for its own energy. If you're going to use amino acid for an energy source, you've got to deaminate it first. The amino group goes to urea. Urea is a non-toxic form, a non-toxic nitrogen waste that can be carried in the blood, sent to the kidneys, and then excreted. So here's another diagram from Kopstead. So post-absorptive state, we're not getting glucose from the gut, but we're breaking down glycogen, and we're performing gluconeogenesis in the liver. That's being sent out to the bloodstream. Glucose is being spared for the brain, while other tissues work on fat metabolism. And now the pancreas is, at first, will reduce its production of insulin and then will produce glucagon to favor these kinds of reactions. So when you have a high blood glucose level, you make insulin. Insulin tells muscle cells to take up the glucose and use it, to make glycogen, to take up the amino acids, to make proteins. Insulin tells the adipocytes to take up the glucose and use it, and turn any excess into fat. Insulin goes to the liver and tells it to take up the glucose to make glycogen, and excess is turned into triglycerides. Triglycerides are packaged in a VLDL and sent to other areas of the body. But if the blood glucose level starts to go down and insulin starts to go down, just the lack of insulin will take us into the post-absorptive state. So muscle will not be taking up glucose. It will be breaking down its glycogen, breaking down its protein, releasing amino acids, and it will be converting to a fat metabolism. Adipocytes will not take up glucose, but will break down triglycerides to release glycerol and fatty acids. The liver will release glucose. It'll break down the glycogen and release the glucose. It will perform gluconeogenesis and release the glucose. This glucose is for the brain. The liver is also going to be taking fatty acids and making ketones. Ketones will go to the body as an energy source for most other cells in the body. Now, this phrase, glucose uptake and utilization, what the heck does that mean? When insulin binds to its receptor, it turns on a second messenger system. It doesn't use a G protein, but it's a second messenger system called a tyrosine kinase. And tyrosine kinase sets off a whole cascade of intracellular events. mTOR is an example of one of the pathway components, and that tells insulin-sensitive cells to insert glucose transporters. When the glucose transporters are then in the membrane, glucose can come in. The first thing that happens is the glucose is phosphorylated. That is the first step of glycolysis. So insulin promotes glucose uptake through the insertion of GLUT transporters and glucose utilization through phosphorylation and putting glucose into glycolysis. So here are some transporters that we think about. Glucose, a GLUT transport, gets glucose into the muscle so we can make glycogen. or use the glucose. Amino acids are used to make proteins. So we don't have a legend here, but the green lines tell you what's happening. The dotted red lines tell you what could happen but are not happening at that time. In adipocytes, glucose uptake, glucose conversion into free fat, into glycerol and fatty acids, which then are made into triglycerides. Triglycerides are going into adipose tissue, but a triglyceride is too big, so lipoprotein lipase breaks it down into a fatty acid and monoglyceride. Those components are taken up and immediately resynthesized back into a triglyceride. Now, the liver, notice there's a black arrow. That's telling us that the liver has a different GLUT transporter than muscle and adipose tissue. And quite frankly, that transporter is there all the time. But during the absorptive state, glucose is entering the liver to be made into glycogen or to be made into fat. So when plasma glucose is high, there are glucose sensors essentially in the beta cells. They secrete insulin. Insulin goes up in the blood. Insulin binds to its receptors in adipocytes and muscle to promote glucose uptake and utilization. And then insulin, the liver, stores glucose but does not break down glycogen, does not perform gluconeogenesis. So if you use the glucose and store the glucose, your blood glucose level goes back to normal. Beautiful negative feedback mechanism. So lots of things stimulate insulin. Your glucose goes up. If your plasma amino acids go up, because insulin is helping you get amino acids into the cell. There is a glucose-dependent insulinotropic peptide, or GIP. There's also a GLP. So this is an example of a hormone called an incretin. When there's glucose in the gut, this hormone goes to the pancreas and says, well, you know, the blood glucose level has not gone up yet, but there's sugar in the gut. So soon there's going to be sugar in the blood. So why don't you make insulin now? That's an example of a feed-forward mechanism. The islet cells are also controlled by the autonomic nervous system. Insulin is inhibited by sympathetic activity, stimulated by parasympathetic activity. Now, during the fasting state, when blood glucose goes down, then insulin goes down, and the post-absorptive reactions start. But when plasma glucose goes down really low, so let's say blood glucose goes down to 80, that will turn off insulin. If it then goes down to like 68, that will turn on glucagon. Glucagon comes from the alpha cells. And the target for glucagon is the liver, the liver, the liver. So the liver will break down glycogen, so there's glucose for the brain. The liver will engage in gluconeogenesis, so there's glucose for the brain. And the liver will take fatty acids and make ketones, so there's food for the other cells of the body. So in the presence of glucagon, the liver will release glucose and ketones. There are other things that influence metabolism during the post-absorptive state. So when glucose is low, that's a stressful thing. We will activate the sympathetic nervous system. We'll have epinephrine from the adrenal medulla, activity of the sympathetic nerves, and that helps bring in lipolysis. Notice glucagon is working on the liver and doesn't really have a lot to do with breakdown of fat. So plasma epinephrine in the sympathetic nervous system, we're going to get glycogenolysis from skeletal muscle. That's also going to promote liver glycogenolysis and gluconeogenesis. But we really need epinephrine in the sympathetics to get lipolysis in the adipose tissue so that we increase plasma glucose but also produce fatty acids and glycerol. Fatty acids then can be used by the liver to make ketones. So here we are back where we started. In the absorptive state, we've just eaten a balanced meal. Most cells of the body are using glucose because it's abundant. And the liver is taking extra glucose and storing it as glycogen and fat. Other cells of the body are engaging in anabolism. They're using glucose as an energy source, but storing amino acids, storing glycerol and fatty acids as triglyceride, and storing glucose as glycogen. In the muscle cell, we would see that. But if you haven't eaten for a while, you want to maintain your blood glucose level, and especially maintain blood glucose for the brain. And there isn't going to be enough glucose for everyone. So how can we manage that? Most cells of the body will convert to fatty acid metabolism, and the ketones from the liver will also be helpful in providing energy. So the liver is producing glucose from gluconeogenesis and glucose from the breakdown of glycogen. So glycogen is being broken down into glucose. That's a catabolic reaction. Triglyceride broken down to glycerol and fatty acids. That's a catabolic reaction. Also, proteins are broken down into amino acids. If we're going to make glucose, we're going to need some amino acids. Glycerol alone is not going to do it. So these will be sent to the liver and involved in gluconeogenesis. So that's an overview and comparing and contrasting of the absorptive and the post-absorptive state. So again, I hope that you are able to view these presentations, and then we will work in class. Dr. Olson has some exercises that we'll think about and to talk about applications of the metabolic states in the human. Thanks so much for listening, and I look forward to seeing you in class.